Okay, so welcome to this video. In this video, what we're going to discuss is desmosomes. Okay, now before I actually define what a desmosome is, I firstly want to talk about desmosomal caterins, because defining what a desmosome actually is quite difficult until you've defined desmosomal caterins. So I want to develop a lot of knowledge, okay, and understanding before we come back to actually what a desmosome is. Okay, right. So, I want to start off by talking about desmosomal caterins. And in order to discuss about desmosomal caterins, we first need to discuss about caterins. And in order to discuss about caterins, we need to talk about cell adhesion molecules. Okay, so that's where we're going to start this video off. We're going to start off with a discussion of cell adhesion molecules. Okay, now for short, cell adhesion molecules are often abbreviated to CAMs. Okay, so C for cell, A for adhesion, and M for molecules. Okay, so C for C for cell, okay, A for adhesion, okay, and then M for molecule. Right. Okay, so cell adhesion molecules then are molecules which are involved in attaching cells together. Okay, right. Now there are two broad types of interaction that cell adhesion molecules can form and thereby attach two cells together. Okay, so one is a direct interaction. So I'll start off by telling you about a direct interaction. So let's say that this is cell 1 here, and I'll colour in cell 1 here in orange. Okay, and let's say that cell 1 is going to have a cell adhesion molecule on its surface. So this square structure here, this will represent a cell adhesion molecule on the surface of cell 1. Okay, so there it is in blue. So this is a cell adhesion molecule. Okay, right. Now, we are going to bind cell 1 to cell 2. So I'll put cell 2 here. Okay, and I'll colour cell 2 in red here. Okay, and basically in a direct interaction between cell 1 and cell 2, what's going to happen is a cell adhesion molecule on the surface of cell 1 is going to interact with a cell adhesion molecule on the surface of cell 2. Okay, so I'll draw this also as a blue box there. So you've got a second cell adhesion molecule on the surface of cell 2, and the cell adhesion molecule on the surface of cell 1 is binding directly to the cell adhesion molecule on the surface of cell 2. The desmosomal caterins that we will see in this video on desmosomes, they are going to interact with each other like so. They are going to be cell adhesion molecules which interact with each other like so. Okay, so this is what's known as a cell-cell interaction. Okay, one cell adhesion molecule on the surface of one cell has bound to another cell adhesion molecule on the surface of the other cell. Okay, let me just, for a complete picture, tell you about the other sort of interaction by which cell adhesion molecules can bind two cells together. Okay, so here's cell 1 again, which we'll colour in orange. Okay, so here's cell 1. Okay, and once again it's going to have a cell adhesion molecule on its surface, so this is a cell adhesion molecule here, okay, which we'll colour in blue. But this time you're not going to have a direct interaction of this cell adhesion molecule with the cell adhesion molecule on another cell. Okay, so we'll draw cell 2 over here, which has a cell adhesion molecule on it. Okay, so here's a cell adhesion molecule on its surface, but these two cell adhesion molecules on the surfaces of the two cells, they're not going to interact directly with one another this time. Okay, they're going to bind to each other indirectly. Okay, right, so let me now show this. So basically, the way we're now going to get these two cells bound together is that these cell adhesion molecules are both going to bind to components of the extracellular matrix. Okay, so let me draw this here. So let's let this green line here represent some component of the extracellular matrix. So maybe it's a collagen fiber, okay, or some proteoglycan fiber. Okay, right. This is going to be a component of the extracellular matrix, and for short I'll denote the extracellular matrix as ECM, but I suppose I should probably write it out at least once. Okay, so this is standing for extra, that's the E, then cellular, that's the C, and then matrix, that's the M. Okay, right, so this is obviously an indirect interaction between the two cells because they're not directly attaching to each other, but they are indirectly attaching the two cells together. Okay, so we now have attached the two cells together. So this is what's known as a cell matrix cell interaction, okay, which just describes exactly what it is. The cell interacts with the matrix, which then interacts with another cell. Okay, so this is a cell matrix cell interaction. Okay, right. So let's now talk about the different families of cell adhesion molecules, because there are 4 superfamilies of cell adhesion molecules. Okay, so the first great superfamily of cell adhesion molecules which cells have on their surfaces are known as the caterins. Okay, and we're going to be very interested in this family because the desmosomal caterins, which are the ones responsible for forming desmosomes, they are within this great family of caterins. Okay, so we're going to have a more in-detailed study of a specific subfamily of the caterins, and we'll come to the subfamilies of caterins in a moment. Okay, we're at the moment talking about the superfamilies of cell adhesion molecules. Okay, so the caterins is superfamily number 1. Okay, then let's go to superfamily number 2. Okay, so superfamily number 2 is what's known as the immunoglobulin superfamily cell adhesion molecules. Okay, right. So, notable examples of immunoglobulin superfamily cell adhesion molecules are, for instance, ICAM1 and VCAM1, which are on the surface of immune cells. Okay, so they're very important in the immune system. Right, so this is the second family of cell adhesion molecules. So that little Ig there, that is short for immunoglobulin. Okay, so the I stands for immuno and the G stands for globulin. Okay, so immunoglobulin superfamily cell adhesion molecules. Okay, so that's the second major family of cell adhesion molecules. Okay, the third major family of the cell adhesion molecules are the integrins. Okay, and the integrins are particularly important in these cell matrix cell interactions, okay, where a cell adhesion molecule interacts with a component of the extracellular matrix. Okay, so they're very important in binding cells to the extracellular matrix, although some integrins are involved in direct interactions where they bind with another cell adhesion molecule. Okay, and the fourth family of cell adhesion molecules are the selectins. Okay, these are also important in immunology, okay, in binding leukocytes, white blood cells, to the endothelium, for instance. Okay, so the selectins is the fourth major family of cell adhesion molecules. Okay, so we're not going to say anything more about the immunoglobulin superfamily cell adhesion molecules, the integrins family cell adhesion molecules, or the selectin superfamily cell adhesion molecules. We're going to focus completely now on the caterin superfamily of cell adhesion molecules. Okay, right. So, in the caterin superfamily of cell adhesion molecules, we can divide it down further into 5 subfamilies. Okay, so I want to now discuss the 5 subfamilies of caterin cell adhesion molecules. Okay, so the first subfamily is the type 1 classical caterins. Okay, right. The most famous caterins are type 1 classical caterins. So, for instance, E-caterin is probably the most famous caterin of them all because of its role in cancer. Okay, it's a type 1 classical caterin. The other famous ones, for instance, N-caterin and P-caterin, they're also type 1 classical caterins. Okay, right, but we're not going to talk about those because those are not involved in desmosomes. Those are involved in very closely related structures known as adherens junctions. Okay, but we're studying desmosomes, which are different from adherens junctions. Okay, right. The next family is what's known as the type 2 atypical cadherins. Okay, again, we're not going to discuss the type 2 atypical cadherins. We're just discussing this so that you've got a broad picture of where the desmosomal cadherins actually fit into this great cadherin superfamily of cell adhesion molecules. Okay, then next up is the star of the show. Okay, this is the desmosomal cadherins, and we will be discussing the desmosomal cadherins in more detail because those are going to be involved in the formation of desmosomes, hence their name. Okay, right. Then the final two families are, we're getting more and more niche basically. The flamingo cadherins, okay, and then also the protocadherins is the fifth family. Okay, and again, we're not going to discuss these two families either. Some people will even say that there is a sixth subfamily of cadherins, and the reason is that actually there are many cadherin proteins, okay, and not all of them actually fit into these five families. So there's then the final family, which are the cadherins which don't fit, basically. Okay, so if you have a cadherin which doesn't fit, you put it into this sixth family, which is all the cadherins which then don't fit. Okay, but we're not going to discuss that family either. So, we are now going to discuss the desmosomal cadherins in more detail, these ones here. Okay, so there aren't actually that many of them, is the nice thing. We're going to talk about all of them. Okay, right. So there are two major families of desmosomal cadherins. Okay, there are the desmoglines, okay, and then there are also the desmocolins. Okay, so we're seeing these continuous subdivisions, basically. We started off with the cell adhesion molecules. We saw that there were these four superfamilies of cell adhesion molecules. We then pressed on the cadherins, and we then saw that there were loads of subdivisions of the cadherins. We then selected desmosomal cadherins, and now we're seeing that there's further division in the desmosomal cadherins. So there are two families of desmosomal cadherins. There are the desmoglines, which I'll have in pink here, okay, and there are the desmocolins, which I'll have in orange here. Okay, so there are four different desmogline proteins. Okay, there is desmogline 1. Okay, and for short, desmogline is abbreviated to DSG. So there is desmogline 1. There is desmogline 2. Okay, there is desmogline 3. And finally, there is desmogline 4. Okay, right, and these are all separate proteins, okay? They are all coded for by different genes, okay? And those genes will have different sequences of organic bases and therefore will code for proteins which have different sequences of amino acids, okay? However, they are all going to have very similar structure, as we'll come to in a moment. Okay, right, now let's discuss the desmocolins. Okay, so there are 3 genes for desmocolins. However, each gene has 2 known splice variants. So I'll start with the 3 genes. There is desmocolin 1. Okay, there is desmocolin 2, and then, by the way, desmocolin is often abbreviated to DSC, like so. So there is desmocolin 1, desmocolin 2, and desmocolin 3, like so. But these are the genes, okay? Each of these genes can actually produce 2 different proteins, okay? So desmocolin 1 gene can produce the protein desmocolin 1A, and it can also produce the protein desmocolin 1B, okay? Desmocolin 2 gene similarly can produce desmocolin 2A and desmocolin 2B. Okay, and then finally, it's very nice, desmocolin 3 can also produce desmocolin 3A and desmocolin 3B. Okay, right, before I explain what the running theme is okay, what the difference between the A's and the B's is. Okay, I'm firstly just going to make sure everyone understands the principle of a gene being able to code for more than one protein. Okay, I want to make sure everyone understands the concept of different splice variants. Okay, so let me just remind you of the central dogma of biology. Okay, so let these two parallel lines that I'm drawing here represent a piece of double-stranded DNA. Okay, so I'll write dsDNA like so to mean double-stranded DNA. Okay, right, and let's say that this portion that I am now highlighting in, well, not highlighting yet, but boxing here. Okay, I'll highlight it now. I'll highlight it in vivid purple here. Let's say that this portion that I've now outlined in vivid purple, let's say this is a gene. Okay, so here we have a gene. Okay, right. Now, the gene will have a coding strand. Okay, and this is the strand that the RNA polymerase enzyme, okay, specifically in humans, RNA polymerase 2 enzyme is going to work its way along and create a piece of mRNA for. Okay, so this is the coding strand. RNA polymerase will always use one of the strands of the DNA to make its mRNA. Okay, and that one that it uses is the coding strand. The other one is known as the non-coding strand. So this is the less important one that's just complementary to the coding strand, basically. So this is the non-coding strand here. And I think I'll colour in the non-coding strand in yellow. Okay, right. So the enzyme RNA polymerase 2, which is often abbreviated to RNAP2 for short, RNA for RNA, ribonucleic acid, P for polymerase, and then 2 for 2. So RNAP2. This is a huge, great complex of a huge number of different proteins. This is going to come and work its way along the coding strand of the DNA, okay, and synthesize a piece of mRNA. Okay. However, this piece of mRNA that you produce, this is complementary to that coding strand. This is not yet ready to actually go for a ribosome. Okay, so here it is here in blue. This is what's known as a piece of pre-mRNA. Okay, and before the piece of pre-mRNA can actually go for a ribosome, it needs to have post-transcriptional modifications occur to it. Okay, and the most notable of the post-transcriptional modifications is a process known as splicing. Okay, so what's the principle of splicing? Well, the principle is this, that this piece of pre-mRNA we have here, also sometimes known as the primary transcript, because it's the piece that you have produced directly by transcribing the coding strand of the DNA. Okay, this piece of mRNA, it's not ready to actually go for a ribosome yet, and one of the main reasons is that there are portions in this piece of pre-mRNA which are not actually supposed to be translated. Okay, so, taking a different tact, let's outline the portions that actually are supposed to be translated. And I'm just doing this arbitrarily, okay, just to make the point clear. So let's say that these portions that I have boxed here, these are the portions that are actually supposed to be translated. Now, the portions that are actually supposed to be translated are known as the exons. Okay, so let's highlight these exons up in orange here. Okay, however, in between the exons, you have portions that are not actually supposed to be translated. Okay, so we have these 4 portions that are not supposed to be translated. Here's the third one, here's the fourth one. And these portions that are actually not supposed to be translated, these are called the introns. Okay, so you are not meant to put this pre-mRNA for a ribosome to be turned into a sequence of amino acids, because you'll get amino acids corresponding to all the organic bases, or all the triplets of organic bases in the introns. And that's not good at all. You'll end up with a dysfunctional protein that will probably just aggregate together and form amyloid plaques. Okay, not good. So, what you need to do is you need to chop the introns out, basically, and re-stick the exons together. Okay, and this will produce you a much smaller piece of mRNA, which I'll colour in orange here. Okay, which just now consists of the exons. Okay, and this process of chopping the introns out and then re-gluing the exons together is known as splicing. And the final piece of mRNA you get here is known as the mature mRNA. Okay, this can now go through a ribosome. It can be translated and turned into a protein. Okay, so it can be translated and turned into a protein. Okay, so that's all well and good. But what does this have to do with splice variants? We were after trying to explain how one gene, for instance the desmocolin 1 gene, can result in 2 different proteins being produced. Okay, how can this gene code for this protein and also this protein? Well, basically, the principle is this. Some genes will produce pieces of pre-mRNA which don't just have one way in which they can be spliced. Okay, some genes do produce one piece of pre-mRNA which is only ever spliced in a particular way, and therefore you always produce the same mature mRNA, so there's no variability in how you can splice it. There is only one option available for the cell's machinery. It can only turn this into this. Okay, however, for some pre-mRNAs it's not the case. Instead, for some pre-mRNAs, there are multiple different options that the cell's machinery has, i.e. there are multiple different ways it can splice up the pre-mRNA. Okay, so let me give you a really easy to understand example of this, and often it's far more complicated than this, but this will get the basic concept across. Okay, so let's say that this final exon here, let's say that this is not essential. Okay, you do not have to include it. You can include it, okay, and therefore you join all 3 of these exons together to produce a piece of mature mRNA, or you cannot include it. Okay, so in the splicing process you have a choice to make. You can either include that or you don't include that. Now, if you include that, you'll get this longer piece here. Okay, but if you don't include that, you'll get a shorter piece now that only consists of 2 exons. Okay, so here is this shorter piece of mature mRNA. So when you have a piece of pre-mRNA, you have 2 options. You can either make this longer piece or you can make this shorter piece. Okay, so therefore there are different mature mRNAs that you can make from the same pre-mRNA, and these 2 different mature mRNAs, they will go on to make different proteins, and these different proteins, those are known as the different splice variants of the same gene. Okay, so that's the basic principle of splice variants, that they result because of different ways of splicing up the pre-mRNA, which will produce different mature mRNAs, and those different mature mRNAs will then go on to make different proteins. So if the cell is producing a huge amount of this pre-mRNA, let's say hundreds of the things, then some of them will go down one route and be turned into one mature mRNA, and others will go down another route, okay, and be turned into a different mature mRNA, and therefore you'll get both of these 2 different types of protein being produced. And that's a very simple example. Some pre-mRNAs can have a huge number of different ways that they can be spliced up, and therefore will be producing a huge number of different proteins when you have multiple of these pre-mRNAs that might all be going down different splicing routes. Okay, right, so that's the principle of how one gene can produce different types of proteins. Okay, that's the principle of splice variants. So each of these desmocolin genes, desmocolin 1, desmocolin 2, and desmocolin 3, the pre-mRNA has 2 different ways that it can be spliced, okay, and these result in different proteins, okay. Right, okay, so let's now actually see the structure of a desmosomal cadherin. Okay, right, so luckily for us, all of these desmosomal cadherins, all 10 of them, okay, we've got 4 desmoglein desmosomal cadherins and 6 different desmocolin desmosomal cadherins. All of them actually have the same basic structure. So let's now have a look at this same basic structure that all the desmosomal cadherins share. Okay, firstly, what I just want to mention is the thing which links all caterins together. So the desmosomal caterins, remember, were just one subfamily of this huge superfamily of cell adhesion molecules known as the caterins. Now, what is the feature that all caterins actually share? Well, basically, you have to have a special domain. Okay, and this special domain that all caterins have is something known as an extracellular, okay, cadherin domain. Okay, and for short, an extracellular cadherin domain is often abbreviated as an EC domain. Okay, right. Now these are just a domain, okay, which many different proteins can have within them, okay, and it's around 110 amino acids. Now, some cadherin proteins have multiple of these, okay, some of them will only have one, but you can actually vary this between 1 and 34, so some cadherins have 34 of these extracellular cadherin domains. For that reason, this is sometimes often referred to as the cadherin repeat domain, because basically it can be repeated up to 34 times in a cadherin protein. Okay, so this is the characteristic structure then that all cadherin proteins have, and that's what defines them as a unique superfamily of proteins, basically. So if these desmosomal cadherins are going to be within this superfamily of cadherins, they better, by God, have an extracellular cadherin domain, at least one of the things. In actual fact, we're going to see that these desmosomal cadherins are all going to have 4 extracellular cadherin domains. Okay, so let's have a look at this. So, right. So all of these desmosomal cadherins, as we've discussed, they are individual polypeptides. Okay, they've all got an individual little gene, and the gene codes for a protein. Okay, yes, some of the desmocolins don't have their own gene because some of those genes code for multiple proteins. But the point is that they are all individual polypeptides. So they're all going to have an amino terminus and a carboxylic acid terminus. Okay, right. So let's let these 2 parallel lines that I'm drawing here represent the cell membrane. Okay, so it doesn't represent a double-stranded piece of DNA anymore. It represents the inner and outer leaflet of the phospholipid bilayer. Okay, so this is the extracellular fluid side, the ECF side, and this is the cytoplasmic side. Okay, so that means that this line here represents the outer leaflet of the phospholipid bilayer, and this layer here represents the inner leaflet of the phospholipid bilayer. Okay, right, and I think we'll actually have a break here, and we'll continue this discussion in the next video, where we'll see the structure that all desmosomal cadherins have.